Phage therapy is a promising alternative to traditional antibiotics, which are becoming less effective due to the rise of antibiotic-resistant bacteria. Bacteriophages are viruses that specifically target and infect bacteria, making them a highly targeted and effective treatment option. The image shows a microscopic view of a bacteriophage attacking a bacterium, highlighting the intricate details and interactions between the two. The vibrant color scheme and bright, clear lighting help convey the scientific precision and potential of phage therapy as a solution to the growing problem of antibiotic resistance. Phage therapy offers a novel approach to treating bacterial infections and could play a crucial role in addressing this global health challenge. Bacteriophages are viruses that infect and replicate within bacteria. They are highly specific to their bacterial hosts. The phage life cycle involves attaching to bacteria, injecting genetic material, replicating, and then lysing or killing the bacterial cell. After replication, new phage particles are released from the infected cell, ready to infect other bacteria. Phages play a crucial role in regulating bacterial populations and have potential applications in medicine and biotechnology. Early 20th century, scientists observed that bacteriophages had the ability to kill bacteria in natural environments. Frederick Twert and Felix de Herelle independently discovered phages in the early 1900s, sparking initial research into phage therapy. Phage therapy gained traction during World War II, especially in Georgia, where it was used to treat bacterial infections. With the rise of antibiotics, phage therapy research declined in Western countries, but continued in Eastern Europe. Recent increases in antibiotic resistance have led to a resurgence of interest and research into phage therapy as a potential alternative. The key advantage of phage therapy over antibiotics is its narrow spectrum. Phages target specific bacterial strains, leaving the beneficial microbiome intact. Another advantage is the reduced risk of resistance. Phages can evolve alongside bacteria, constantly adapting to overcome resistance mechanisms. Phages also tend to have lower toxicity compared to antibiotics, as they are naturally occurring viruses that generally have minimal side effects on the host. These features make phage therapy a promising alternative to traditional antibiotic treatments, especially for drug-resistant infections. Phages are viruses that infect and kill bacteria. They do this through a specific four-step process. First, the phage binds to receptors on the bacterial surface, attaching itself. It then injects its genetic material, either DNA or RNA, into the bacterial cell. Inside the cell, the phage hijacks the bacterial machinery to replicate itself, producing many new phage particles. Finally, the new phages cause the bacterial cell to burst open, or lyse, releasing the progeny phages to infect more cells. Phages, or viruses that infect bacteria, have an incredible ability to target specific bacterial strains. This specificity comes from the interactions between the phage's tail fibers and receptors on the bacterial cell surface. This specificity is critical for using phages as targeted treatments for infections. By carefully selecting phages that target the specific bacteria causing an infection, we can develop personalized phage cocktails. This targeted approach helps minimize damage to the beneficial bacteria in the patient's microbiome. It also reduces the risk of antibiotic resistance and side effects compared to broad-spectrum antibiotics. Environmental samples are collected from potential phage sources, such as soil, water, or sewage. Bacteria are grown in the lab, and the collected samples are introduced to create an enrichment culture. Individual phages are isolated using plaque assays, which allow us to identify and purify specific phages. We then characterize the isolated phages, determining their host range and lytic activity to identify those with desirable therapeutic properties. Finally, we analyze the phage's genetic makeup through genome sequencing, which can reveal potential applications for the phage. This multi-step process is crucial for identifying phages with the right characteristics for therapeutic use. Phages, or viruses that infect bacteria, are constantly evolving to overcome bacterial resistance. By using a cocktail of different phages, each with unique mechanisms of action, we can reduce the likelihood of bacteria developing resistance. Phage engineering allows us to tailor phages to target specific bacterial strains or enhance their effectiveness. Combining phage therapy with antibiotics or other treatments can create a synergistic effect, further combating bacterial resistance. Regulatory approval is essential for ensuring patient safety with this new field of phage therapy. Phage strains can be difficult to standardize, which poses challenges in developing reliable and consistent treatments. More research is still needed to fully understand the long-term effects of using phage therapy. Cost-effective large-scale production of specific phages is crucial for making this treatment widely available. Phage therapy is a promising treatment approach, but it faces unique regulatory challenges compared to conventional pharmaceuticals. Regulatory agencies are still developing specific guidelines for phage therapy, so we need to carefully navigate the current pathways. Rigorous testing is essential to demonstrate the safety and efficacy of phage treatments. Just like any other medical intervention, we must ensure phage therapy meets the same high standards for safety and efficacy as other approved treatments. This is a complex regulatory landscape, but with careful planning and execution, we can successfully bring phage therapies to patients. Clinical trials are crucial for evaluating the efficacy and safety of phage therapy in human patients. These trials help establish the effectiveness of phage treatments for different types of infections. Case studies offer real-world examples of successful phage therapy, demonstrating its potential to treat drug-resistant infections that are otherwise difficult to manage. The case studies provide valuable insights into the practical application of this innovative approach. Together, clinical trials and case studies are essential for advancing our understanding and adoption of phage therapy as a viable treatment option. Antibiotic-resistant infections are a growing concern, posing serious threats to public health. Infections like MRSA and CRE are very difficult to treat with traditional antibiotics. Phages offer a potential solution by targeting specific bacteria, even those resistant to antibiotics. Phages can be tailored to specific strains of bacteria, reducing harm to beneficial microbes. Phage therapy is showing promising results in treating antibiotic-resistant infections, offering new hope for patients with limited treatment options. Skin infections caused by antibiotic-resistant bacteria can be treated with phage therapy, including acne, boils, and wound infections. Phages can effectively target bacteria that often infect burn wounds, reducing infection risk and promoting healing, which is especially beneficial for severe burn patients. Diabetic foot ulcers are prone to bacterial infections, and phage therapy offers a potential alternative to antibiotics that often fail to penetrate deep tissues. Phage therapy can target specific bacteria involved in dental infections, such as periodontitis, providing a targeted and localized approach to treatment. Phage therapy is a promising approach for treating gastrointestinal infections. Phages, or viruses that target bacteria, can selectively eliminate harmful bacteria in the gut. This leaves beneficial microbes untouched, preserving the healthy gut microbiome. Eliminating pathogenic bacteria can also help reduce inflammation in the gut. Alleviating inflammation can ease symptoms like diarrhea and abdominal pain. By restoring balance to the gut microbiome, phage therapy promotes overall gut health. This makes it an attractive alternative to traditional antibiotics for GI infections. Phage therapy is a promising approach for treating respiratory infections, as phages can effectively target bacteria in the lungs. Phages can be administered intravenously to reach the lungs, allowing for targeted treatment of respiratory infections. Phages can be delivered to different areas of the respiratory system, enabling targeted treatment of various bacterial infections. This slide provides an overview of how phage therapy can be used to address respiratory infections by targeting the lungs and using intravenous delivery. Chronic wounds that are resistant to antibiotics are a major challenge. Phage therapy is an emerging approach to treat these difficult-to-heal wounds. Phages are viruses that can specifically target and kill bacterial infections. Applying phages to wounds can help prevent bacterial infections, especially when antibiotic resistance is a concern. This is an exciting new area of research with the potential to improve wound healing and patient outcomes. Personalized phage cocktails are a novel approach to treating bacterial infections. These cocktails are tailored to target the specific bacterial strains causing a patient's infection. This individualized therapy addresses the unique characteristics of the infection, leading to more effective treatment. Different phages can effectively combat various bacterial species, allowing us to address the diversity of bacteria that may be present. The combination of these targeted phages in a cocktail provides a powerful tool for fighting complex infections. Phage therapy is a promising solution to treat bacterial infections in animals, especially as antibiotic resistance continues to grow in livestock. Phages can be used to control outbreaks of foodborne pathogens like E. coli and Salmonella in livestock, helping to improve food safety. Phage therapy is also applicable for treating infections in pets, such as skin, urinary tract, and respiratory infections. Using phages can help reduce the need for antibiotics in animals, which is important for combating the rise of antibiotic resistance. The image shows a friendly veterinarian among a group of farm animals, highlighting the potential of phage therapy in livestock and pet health. Phages can selectively target and kill harmful bacteria in the microbiome without impacting the beneficial bacteria. Phage therapy may actually have a positive effect on the overall balance and health of the microbiome by promoting the growth of beneficial bacteria. Using phage therapy could be an effective way to restore the proper balance and diversity of the gut microbiome, which is so important for overall health. The vibrant, colorful image of the complex microbiome highlights the intricate ecosystem of bacteria, viruses, and phages that make up this crucial system in our bodies. Phage production is a multi-step process that requires careful optimization and scaling up. Selecting the right phage strains is key. We need to identify high-yield strains that can be reliably produced at scale. Large-scale bioreactors are used for the actual phage fermentation process. Purification is critical to remove bacterial debris and other contaminants, ensuring a high-quality final product. Formulation development is necessary to create stable phage preparations for storage and delivery. Phages are commonly formulated as liquid solutions, often in buffered saline, for intravenous or oral administration. Lyophilization, or freeze-drying, is used to stabilize phages, extending their shelf life and allowing for easier transportation and storage. Encapsulation in nanoparticles or other carriers can enhance phage delivery to specific tissues or organs, improving therapeutic efficacy. Combining phage therapy with other antibiotics or immunotherapies can enhance effectiveness and address antibiotic resistance. The combination of phage therapy and antibiotics can have a synergistic effect, where they work together to more effectively eliminate bacteria. Phages can weaken the defenses of bacteria, making them more susceptible to the action of antibiotics. Using a combination of phages and antibiotics can help overcome antibiotic resistance, as phages can target specific bacteria while antibiotics have a broader spectrum of activity. This combined approach expands the treatment options for bacterial infections, including those caused by multi-drug-resistant bacteria. The images on the slide show a close-up of a lab experiment with petri dishes containing phages and antibiotics, as well as a scientist examining the results in a sterile lab environment. Phage engineering and synthetic biology offer exciting possibilities for improving the effectiveness of phage therapy. By engineering phages to target specific bacterial strains, we can enhance treatment efficacy while minimizing disruption to the beneficial microbiome. Modifying phages to withstand harsh environments like the human gut can enhance their effectiveness in treating gastrointestinal infections. Engineering phages to produce more potent toxins or enzymes increases their ability to kill targeted bacteria. Combining engineered phages with antibiotics or other antimicrobial agents can enhance treatment efficacy against multi-drug-resistant infections through a combinatorial approach. Phage therapy is a promising alternative to traditional antibiotics, but its regulatory landscape is complex and evolving across different countries. Regulatory frameworks for phage therapy vary, with some nations having more developed approaches than others. Global regulatory bodies are working to establish specific guidelines for phage therapy, addressing safety and efficacy concerns. Countries are collaborating internationally to standardize regulations and promote global research and development in this field. The regulatory landscape highlighted on the map shows where the most developed frameworks for phage therapy currently exist. This collaborative effort is crucial to unlocking the full potential of phage therapy as a powerful tool against antibiotic-resistant infections. Phage therapy research requires significant investments to isolate and characterize phages, so patent protection is crucial for commercial viability. Commercialization often involves licensing agreements with pharmaceutical companies or collaborations with research institutions to scale up production and distribution. Phage therapy holds immense global potential, but navigating the regulatory landscapes in different countries is crucial for achieving widespread market access and impact. Phage therapy is an emerging treatment that uses viruses to target and kill specific bacterial infections. It's important that patients fully understand the experimental nature of this treatment and the potential risks and benefits. The informed consent process is critical. Patients must have clear documentation outlining the details and any potential complications. Phages are highly specific, only targeting certain bacteria. This raises ethical considerations about the impact on the delicate balance of the human microbiome. We need to carefully consider the broader effects phage therapy could have on the body's natural bacterial ecosystems. Phage therapy is a promising approach that involves using viruses to treat bacterial infections. Bringing together scientists, clinicians, and industry partners is key to addressing the challenges in this field. Expanding clinical trials is crucial to assess the efficacy and safety of phage therapy. Developing new phage isolation methods and engineering techniques can drive technological advancements. Securing funding is essential to support research, development, and clinical trials in engineered phage. We can tailor phage properties to enhance their efficacy and specificity, potentially targeting specific bacterial strains or overcoming resistance. Combination therapies. Combining phage therapy with antibiotics or other treatments can achieve synergistic effects, potentially boosting efficacy and reducing resistance. Personalized phage cocktails. Developing individualized phage cocktails tailored to the specific bacterial infection can enhance treatment outcomes. Advanced delivery methods. Exploring innovative delivery strategies, such as targeted drug delivery systems, can improve phage penetration and efficacy. Phage therapy has faced regulatory hurdles, as existing regulations are primarily designed for traditional antibiotics and may not adequately address phage therapy's unique characteristics. Scaling up production to meet the demands of widespread use is a significant challenge, and standardization of phage production processes and quality control measures are essential for ensuring consistent efficacy and safety. Securing investments from pharmaceutical companies and government agencies is crucial to advancing clinical trials and making phage therapy more accessible. Educating healthcare professionals and the public about the potential benefits of phage therapy is essential for its wider acceptance, as addressing public concerns about its safety and efficacy is crucial. Phage therapy holds significant promise as a treatment for antibiotic-resistant infections, which are a growing global health concern. Continued research is crucial to overcome the remaining challenges and ensure the safe and effective application of phage therapy. Collaboration among researchers, clinicians, and industry stakeholders is essential for the successful implementation of phage therapy. The development of clear regulatory frameworks for phage therapy is crucial for its wider acceptance and adoption by the medical community.